Penicillin. How penicillin kills bacteria. Penicillin kills bacteria by interfering with the ability to synthesize the cell wall. The inappropriate use of antibiotics, prophylactic treatment of herds in meat production, and pointlessly treating viral infections with antibacterials floods our environment with drugs that select populations of antibiotic-resistant bacteria. Although penicillin was a wonder drug when discovered, some bacteria exposed to penicillin survived because they produce the enzyme B-lactamase that destroys penicillin's structure. A solution was to devise other penicillin-like antibiotics, B-lactams, with structures that are not destroyed by B-lactamase. Methicillin is one of those drugs. Unfortunately, an increasing threat is now methicillin-resistant Staph aureus, or MRSA, which avoids methicillin and other B-lactams by an altercation in the MRSA's penicillin target binding site. Bacteria are constantly evolving such new defenses, so treatment of bacterial infections require a constant search for antibiotics with novel modes of action. MRSA produces a penicillin-binding protein, 2A, mediated through the MECA gene, which is carried on the staphylococcal cassette chromosome, MEC, SCC-MEC, of which there are at least 6 different types recognized, and this results in resistance to all beta-lactam antibiotics. There is much debate on whether strains of MRSA are intrinsically more virulent than methicillin-susceptible isolates, but it is agreed that MRSA causes the same range of infections, resulting in excess healthcare costs, prolonged hospital stays, and significant mortality. MRSA is endemic in hospitals globally, except in Scandinavia and in the Netherlands, although declining rates of MRSA BSI have been seen in recent years in the UK, France, and other European countries. Vulnerable patients, particularly at risk, are those who have undergone major surgery and patients in the intensive care units. Although 50 to 60% of patients with MRSA are merely colonized, representing asymptomatic carriage, serious infections occur such as BSI, respiratory tract, and bone joint infections. These infections are then more difficult to treat than infections caused by methicillin-susceptible isolates, and MRSA can spread easily among patients in the hospital. Beta-lactam antibacterials bind to several penicillin-binding proteins in bacteria. Some of these proteins are transpeptases, which are required for cross-linking in the peptidoglycan layer of the cell wall surrounding certain bacteria and are essential for their survival. Inhibition in transpeptase activity by B-lactam antibacterials prevents the bacterium from synthesizing an intact cell wall when it divides. The transmembrane osmotic gradient then leads to swelling, rupture, and death of the bacterium. Some bacterial cells also contain enzymes that cause cell lysis when activated. The binding of B-lactam antibacterials to other specific penicillin-binding proteins within bacteria reduces the production of natural inhibitors of lysis-inducing enzymes, promoting lysis of the bacterial cell wall. Enzymatic inhibition mechanism of penicillin. It does this by mimicking the D-enanyl and D-alanine residues that would normally bind to the site. Penicillin irreversibly inhibits the enzyme transpeptase by reacting with a serine residue in the transpeptase. This reaction is irreversible, and so the growth of the bacterial cell wall is inhibited. And this is the bound structure of penicillin.